Chapter 17 of our textbook, which was on learning and memory. Segment 3 specifically started talking about the neural mechanisms of memory storage. So the cellular and physiological bases of memory, which the text says is also called engram or memory trace. So new learning involves changes in the strength of existing synapses and biochemical signals that may produce those changes. So an important vocabulary term to keep in mind for the rest of this portion of the textbook is neuroplasticity, which is the ability of the nervous system to change in response to experiences in the environment. And this is found in nearly all animals, which suggests that this is an ancient and vital product of evolution. So the first important section is memory storage requires physical changes in the brain. And so this section talks about synaptic alterations and how they might be the basis of learning. So plastic changes at synapses can be physiological or structural. So changes such as a greater release of neurotransmitters, a greater number of receptors, or increased sensitivity of those receptors, all these changes can alter in size of the postsynaptic potential. And synaptic activity may also be modulated by inputs from other neurons as well. And so new synapses may form or synapses may be eliminated as a function of training, which may also lead to the reorganization of synaptic connections. So the next major component of learning is varied experiences in environments, and these experiences can cause the brain to change and grow. So living in a complex environment produces biochemical and anatomical changes in the brains of rats, as found through studies. Standard studies involve putting the rats in one of three conditions. So the first one is standard condition, where they are housed in small groups in standard laboratory cages. The impoverished conditions involve them being housed individually in standard lab cages, and enriched conditions involve putting the animals in large social groups in cages with toys and features, and this provides enhanced opportunities for learning. So through these studies, it was found that plastic changes in the brain are linked to the environmental enrichment, that is the enriched condition. So the enriched condition animals have heavier, thicker cortexes, they show more cholinergic activity, and their cortical neurons have more dendritic branches compared to the impoverished condition animals. And MRI scans of human brains show similar results, that adolescents from advantageous socioeconomic environments have thicker cortexes than adolescents from lower socioeconomic environments. So instead of trying to find exact synaptic changes that underlie specific instances of learning, since there are too many synapses in human brains, researchers study simple learning in simple animals. The next major section is invertebrate nervous systems and how they show plasticity. The text specifically talks about the study of aplasias, which are sea slugs with simple nervous systems that are capable of non-associative learning, which is a simple type of learning where a single stimulus is presented once or repeatedly, and has three major forms: either habituation, which is a decrease in response to a stimulus as the stimulus is repeated; dishabituation, which is the restoration of response amplitude following the habituation, or sensitization, which involves the organism becoming more responsive to most stimuli, not just that one, after being exposed to an unusually strong or painful stimulation. So with the sea slugs, their siphons were squirted with water repeatedly, and eventually they retracted their gills less and less as they learned that the water will not have hurt its gill. And the habituation was caused by changes in the synapse between the sensory cell that detected the squirt of water and the motor neuron that retracted the gill. And so the gill stopped retracting after less and less of the neurotransmitter was released. So strong stimulation on the skin causes stimulation of the siphon to produce larger gill withdrawals. And similar findings of studies with more diverse species provided evidence that information can be stored in the nervous system by changes in the strength and the number of synaptic contacts. So the next section of the text talks about the cerebellum and its implicated involvement in simple learning in animals. So while the sea slugs had a fixed circuit for the learning in the study, mammals' brains have billions of neurons that are definitely not as organized as those of the sea slugs. So this led researchers to study classical conditioning, from which they found the cerebellum to be important in. So a study done with rabbits involved air being puffed into their corneas, and thus they blink. And so the unconditioned stimulus, the air puff, follows a tone, which is the conditioned stimulus, and eventually the rabbit came to blink at the tone, the conditioned stimulus, even if it wasn't followed by the air puff. And so this led to a conditioned response. So in the study, researchers implicated a cerebellar circuit to be necessary for conditioning to aversive stimuli. So the interpositus nucleus, which is a region in the cerebellum that receives climbing fibers, axons, from the brainstem neurons that send information about both the corneal stimulation from the unconditioned stimulus and the auditory information from the conditioned stimulus, the tone. And so the information from the unconditioned stimulus, the air puff, and the conditioned stimulus, the tone, both converge in this nucleus. So drugs that block GABA's effect, or antagonistic drugs to GABA, work by shutting down the interpositus nucleus. And so GABA antagonists cause the conditioned behavior to disappear and prevents the conditioning from occurring if injected before the process of the classical conditioning even takes place. And so the eye blinking conditioning study is consistent with other studies on human participants as well. And so studies have also, and lastly, implicated the cerebellum as a crucial brain region for conditioning across other domains, such as emotions, fear, and cognitive learning as well.